let's move on to the section 3, field 2, with the group of bitter gourd in food and nutrition. We are going to have 4 person is going to be Dr. Bhaskarachary, scientist C-level of Indian National Institute of Nutrition. He has a very vast research experience in several fields including food chemistry, metabolic diseases, bioavailability, etc. And I request him to give his presentation.
Thank you, madam. At the outset, my sincere thanks to the organizers who invited me to give an experience on bitter gourd to this august audience. We are talking about the bitter gourd since morning, and I am going to focus the real data of from the National Institute of Nutrition's perspective and the data generated, and this is the disease prevalence. Actually, in India, 5 to 15% urbans are of having diabetes mellitus, and semi-urban 4 to 6. Even in rural populations, 2 to 5%. And there was a huge discussion about this because India is going to be the capital for the diabetes soon, and more so if you look at the figures of this of Hyderabad is going to be the capital for the world diabetes very soon. When people are thinking about when scientists don't know any reason, then they will simply run away with the genetics usually. But if the real genetics are the factor, or the genes which we are wearing outside is the matter. Really the matter is what we are wearing outside the body. The genes are mostly influencing your diet and lifestyle is the predominant factors for passing the genetics. Actually, if you could see it, I am a son of an Ayurvedic physician and father of 2 budding allopathic doctors, that I have a good experience of both these allopathy as well as Ayurveda, where I could see in my father's practice, I hardly see any diabetics when I was in my childhood, but now you could see it is flooded with these people. And look at the hypertension of this country, particularly every 4th urban Indian is hypertensive. The predominant reason is again about your diet and the lifestyle is the basic thing that one should really look at. This data of from various ages also between men and women, and approximately of 25% of urban Indians are hypertensive. And look at the production and consumption. Since morning I am listening about the bitter gourd, but on the overall perspective you could see the total production of fruits in the world around 370 million metric tons. India ranks first in the world with an annual output of so and so. India is also the 2nd largest producer of the vegetables in the world and accounts for about 15% of the world's production. Including the population also, it's an explosive way. India is going up next to China. But if you look at the consumption pattern, as somebody was rightly mentioned in the early morning, that the consumption of bitter gourd, what could be the level really. But if you look at the recommended dietary allowances from the National Institute of Nutrition and look at the figures which are consumed, and particularly which we are interested today about the other vegetables where your bitter gourd also comes, this even we are not meeting the recommended dietary allowances. An average Indian is eating of the vegetables, and then we are now talking about the bitter gourd, and many other gourds are also not really including in our diet. Probably that could be the reason. When I have focused about the diabetes and the hypertension, my intention is about this bitter gourd is very rich source of potassium, which is very useful against the hypertension, as well as of many factors which influences the glycemic ranges in the body. And bitter gourd was used in Vedic times, particularly Aryans used for this for medicinal purpose in addition as a vegetable during 2000 to 200 BC. Very long back the history says that India has used this, and in Sanskrit also it has been named and it has been changed also the names. And for present work I have taken, after I got a call from this conference that there's a conference that I have to post a real data rather than a review of literature. Then we have collected locally available in the Hyderabad and Secunderabad markets, Actually, I'm also doing the national sampling for this country. Basically from the all over the 107 points across the country from Jammu Kashmir to Kanyakumari, we are collecting the samples, we are generating the nutrient database, and my lab is now have an expertise of generating 200 parameters in any given food sample, and probably in by this year end we are going to release the data also for public very soon, which is on par with USDA database. And these are the varieties which I have collected for this work, bitter gourd of various stout variety and long slender variety and very small variety. And you could also see this is the bitter gourd wild tiny varieties where actually tribals bring it to this city. Basically, particularly in the winter season, the markets are flooded with this variety. And then when we are looking at the various factors, and we have seen this because since morning also I think somebody has raised about this bitterness principle. But in Indian culture particularly, if you look at the Ayurveda, in Ayurvedic this thing, a doctor, what the general perception is, what tastes to your tongue, basically, if it tastes bad, that's good for your health, they used to tell. What tastes to your good, like sugar, is bad to your health. This is a general concept which they will advocate in this country. In that perception, this has been quoted even in Sushrutas and Charakas also, this bitter gourd particularly. And when people have a problem of eating this because of the bitter principles, and there are methods also have been developed how to mask this bitterness, not to remove this bitterness, but there are methods also to remove the bitterness at a laboratory scale, but you have to mask the bitterness, as somebody rightly pointed out from Germany. It is actually your receptors and your tongue taste buds which will actually, once it passes there from the esophagus, doesn't have any matter for the intestine, where the pH will be 1 to 2, everything will be smashed. So at that level, even to mask for that few seconds or few minutes which is in the buccal cavity, the food, so we can mask that by adopting, particularly I am talking about this region because I am presenting the data of Hyderabad region. So where this practice of adding these Karela, this bitter gourd in the curd, they will put for 20 to 30 minutes and then they will cook. So that bitterness will be masked, not that bitterness will be removed, it will be masked. And they will also use tamarind, also another technique actually in this part. And these two techniques which they can mask the bitter principles, but they will have an added advantage. So how this can be used, really whether these are perception or what would be the real nutritional science when we have, and we have made recipe also out of this. Usually what could be the, because I have a big group of which I am working with the diet and diabetes also, and we have checked this city which is predominant, more than 16% of this city is diabetic people, and we have captured the information that majority of the diabetics in this city are interested to eat bitter gourd because they have some knowledge. Somewhere they have read about bitter gourd, they will also talk about neem leaves, they have talked about bottle gourd and many other things which we have tapped the information, and we are checking whether they are really useful for the hypoglycemic situation in the body. So these are the recipes as it is, how the people of Hyderabad and Secunderabad and the rural places around this state which we have captured, and we have standardized this recipe, and we have done the analysis for this, how this is going to be. And these are the 4 varieties which I have shown you earlier. And when it comes to the component analysis, and as I have told you, my lab actually, we are looking at the different components, not just only carnitine or about a particular compound. See, I have a different opinion when we are discussing about the bitterness of expression also, but you have to look at the different molecules, their combination, their permutation, and my previous speaker talking about the accessibility, affordability, but I am going to talk about bioaccessibility and bioavailability. Whether they are really bioaccessible to your intestine epithelial cells and further bioavailable to the target tissue wherever it is going really, that matters, and what molecules, what combination, what permutation is more important than of an expression of a quality parameters. So these are the various carotenoids which we have screened these bitter gourds, and using the modern methods of analysis of HPLC, and we found particularly these varieties, if you could look at, have, for the sake of presentation purpose, I have put both ripen in the sense, in my view, that becomes totally red when the fruit, what is a raw and ripen. Raw is what we call it as, here we are talking about as fruit, but for the sake of this presentation, when I am talking as ripen, means it is totally changed in its color, green color, that's a chlorophyll. This chlorophyll has been now turned into a different color, where the anthocyanins and the carotenoids which are emerged, and then the composition itself will change. And if you eat such ripen, somebody was asking in the morning also about what could be the nutritional profile of these things, and ultimately what could be the bioavailability, which I am interested. So these things, if you look at from raw to ripen, look at that. Everywhere, particularly the beta-cryptoxanthin, this is xanthophyll oxygenated carotenoids, and some of the oxygenated carotenoids which I am focusing here is the beta-cryptoxanthin, particularly very useful, particularly gut health, intestine health. And this lutein and zeaxanthin is another components which are also present. They are very useful for age-related macular degenerative disease. These two pigments are concentrated in the macular region of the eye, where the blue light will be filtered so that the vision will be, the diminution of vision will be prevented in the aging population. More than 60 years, where you could see these two pigments were actually the concentrations are goes down in the human body. So if you take such vegetables rich in these xanthophylls, oxygenated carotenoids, so that you can postpone your age-related macular degeneration. So in all the ripen samples, these xanthophylls are going up. Look at the viroxanthin is another compounds also. But as a nutritionist, I am interested in beta-carotene, which is a vitamin precursor, converts to vitamin A in the body, the beta-carotene which is present. But after reading this conference, then I thought I should have the real bioactive molecules, which are really biologically active, maybe nutritionally, maybe at a different perspective, but looking at as a singlet oxygen quenchers or free radical quenchers. These molecules are very powerful, which are abundantly present in these bitter gourd samples. And look at this viroxanthin also. And lutein, just now which I have mentioned, which is very rich source, which is very good for particularly, it's a very powerful antioxidant, and which is concentrates in the human macular region, that is in the retina, where it will protect from the blue light which comes from the sunlight. And this is about the total carotenoids, what it will be, not only the both xanthophylls and other things. Another interesting group of molecules in the bitter gourd, or for that matter now the people have interest of the signature molecules in the various vegetables, more so of the bitter gourd. So now in my lab, our target is 100 molecules in single chromatographic run, whether I could look at the signature molecules of these things. And so far now I am successful using the sub 2 micron particle size technology of HPLC. It's not a normal HPLC, it's a fast HPLC, what we call, where you could do within 20 minutes time, you could able to do more than 30-40 compounds with a good precision and accuracy, and with good resolution. I could able to detect these molecules which are abundantly present in these things. So then other interesting thing is, when we are talking about two issues, when we are talking about bitter gourd. One, we are talking for the, particularly for the diabetes, but how it could reduce the glycemic response. And the second aspect is when people are interested in the cancer, also particularly because of the oxidative the load in the body, and how this could be minimized by scavenging these free radicals. By using various methods, I have screened for these varieties of bitter gourds and looked at the ABTS, DPPH and FRAP method. These are the various methods. Actually, there is a lot of discussion also worldwide is going about the antioxidant activity, what method one should really read. Because in the human body, if you could see it, there were many free radicals which will be generated. There will be oxygen derived radicals, there will be nitrogen derived radicals. So not all the radicals will be bombarded with these molecules. They are specific for a particular this thing, or site specific also. That's why sometimes even if they are abundant in the vegetables, whether they are bioavailable and whether it is reaching to the target tissue is also very important when you are talking about the particularly for cancer and for the oxidation reduction studies. So we have screened these varieties and I have ranked them based upon, because both you will have the free radicals also. When you talk about antioxidants, both they are hydrophilic and hydrophobic in nature. Whether they will be soluble in water or whether they are not soluble in water. Based upon that, various essays which we have done, and I have ranked these varieties of these 4 varieties which I have collected. And also what I did, as I have told you, that I have bitterness removed, means, I'm sorry, it has wrongly put there, I have masked this bitterness, in the sense by dipping these bitter gourd samples in the curd. So it's only masking, it's not really removing the bitter principles. So, and then cook with bitterness, so without adding any of these things, what will happen to these things like that, then totally it is ripened when the color changes. If you put it at 24, 48, 72 hours also, now we are studying further on these things and looked at these things of various essays and ranked and overall ranks also which we have given. But the thing is, when I could see it, if you could see, most of the times in the ripen, particularly the wild variety which we are interested, where the people, they are not using any fertilizer or anything, a particular tribe in this part of the country, which they will collect these samples, they don't grow really, it grows wildly, this particular variety, and they will sell in the market. Particularly in the winter season, Hyderabad, Secunderabad markets are filled with this variety. People, they will pay, somebody was mentioning about 8 rupees, 10 rupees, this will go up to 60 to 100 rupees kilo also during that season. And then we have screened, it's a very powerful for antioxidant purpose when we have screened these varieties. And total polyphenols and bioaccessibility. When we are talking, okay, these are abundantly rich molecules, whether they are really bioavailable. When we are talking molecules are in milligrams, if it is so, then there should not be any cancer really, if it is true that it is crossing the membrane and reaching the target tissues and deactivating these molecules. But when we have screened, these are all in vitro studies which we have done. We are going to do the in vivo also, both in animal as well as the human model which we are now planning. But at current data which I am presenting here is of all in vitro studies. And these studies, if you could see the percentage bioavailability, it's nothing more than not even one variety of these things, whether it is removed or whether with bitter, it is, if you could see from even 10% to 25 to that percentage, 10 to 25% the bioavailability. That means 90 to 75, if you could say, it's not really absorbable, these molecules. So that means the absorption, there must be some problem. There must be a competitive inhibition. So these are the things probably which we have, it has triggered us. In a way, this conference helped me to probe further into this, that what combinations probably it is pushing this absorption, whether a particular combination of various molecules will push further or inhibiting, also which we are now looking at. That if you could see it, this is what I have put it pictorially so that you could easily understand the total polyphenols of this kind, but absorption is of that red bar, if you could see it. They are very poor in absorption. And when we are talking about, I have seen the data, I have recently published that total polyphenols which we are talking, which in my view there is no meaning, because we are targeting all the times as gallic acid equivalents or catechin equivalents or ferulic acid equivalents or quercetin equivalents, but these are billions of molecules and we are taking one standard and we are putting it as equivalents, which has no meaning, which I have shown in my recent paper. So taking into that further, looking into individual molecules of these polyphenols, whether there are having any differences in absorption. That also we have tested for the individual, for example, like chlorogenic acid, coumaric acid, ferulic acid, protocatechic acid, vanillic, gallic, like this so on, around 30 molecules so far. Now we have started screening, and particularly with bitter gourd, at least 10 to 15 molecules I have screened already in the in vitro, particularly for individual molecules also in addition to the total. But if you look at chlorogenic acid, if you could see it, percent availability, so there are very poor also, some of them, very poor. So when we are, see, when people did in vitro models and they had shown this is a very powerful antioxidant, then market also going in that way. But whether they are really bioavailable, this is also matters. So what makes this more bioavailable is an interest for a nutritionist, because it should reach the target tissue. So whether it is your method of processing or combination of different aspects of it, which matters for the absorption, which we have further probed also. So these are the various varieties for the various molecules which I am showing it here, bioaccessibility. Then another thing, what I have focused so far about the individual group of molecules together and individual molecules, how they are their absorption. Now we are looking at the different combinations also, which I am not focusing here. But another important is about the glycemic index. Now I have not focused because this country which I have told you is going for a world diabetes, which I am telling about the capital, and most of this city, 16.5% population of this Hyderabad are diabetic. So when we have screened recently, not only the consumption of less of these vegetables, but also the rice, particularly the rice, which Hyderabadis are eating more than 10% polished. So we are almost eating like a starch capsule. So when the starch is loaded, your glycemic response is also going up like anything. The glycemic index studies so far mentioned particularly for the hand pound rice or brown rice are to a lower extent, to some extent something like 0 to 5% polish. But now what is available in the market is 5 to 15% polishing. So that the starch capsule only which we are eating, so that the glycemic response is also going up, so that this bitter gourd will come into the picture. Then how we can counter when people are habituated to eat this jasmine color of this rice in this country, then how to bring down this glycemic index, which we have tested with this bitter gourd varieties. Look at this glycemic index. Normal bitter gourd, if you take the variety, stout variety which I have seen, you see it this bar and this bar. What we have discussed in the fruit here, and I am talking about the ripen means totally the color changed, it becomes fruit, the color which has changed. The glycemic index has increased from, actually bitter gourd is somewhere in the median glycemic index. Actually it will be between 55 to less than 70, the glycemic index is a median glycemic index for the bitter gourd. But when we have made recipe as per the culinary practices of this city, that a little bit jumped really, because of other ingredients which we have used, but still they are in the median range only. With bitter gourd recipe are still in the median range for as a glycemic index. But the totally ripen, when it has become total fruit, color changed, because of other factors which I will also show you what made this bitter gourd to become more glycemic index. So this the four varieties, if you could see it, they are in the median range only. Look at this, what are the factors which will influence basically the glycemic index? The other factors which are contributing for the glycemic index. Resistant starch, which is also in addition to the dietary fiber in the food, which will help to slower the absorption of the glucose. Resistant starch is also another component which also helps to slower the absorption of glucose. And if you could see it, there will be a clear difference between the raw to ripen variety, the resistant starch. So also another important thing what our lab is doing, particularly with the bitter gourd, is alpha amylase inhibition. When we take the starch or the rice or any component, and in the intestine the enzyme tyrin alpha amylase starts right from your mouth, then in the intestine particularly, then the starch will be degraded, and then it will be gone to the monosaccharides, and then it will be absorbed. So amylase and these enzymes are very important for starch breakdown in the intestine. If this is inhibited, so that the absorption of the glucose will be slower, lower, so that the glycemic response also will be slower. Actually, this is the principle of some of the molecules which are abundantly present in the bitter gourd having the alpha amylase inhibition activity. So the polyphenolic compounds which we have screened having the alpha amylase inhibition which we have tested, these are the alpha amylase inhibitors basically which are present in the bitter gourd, which are inhibiting the alpha amylase not to convert the starch to the glucose, so that the time lapses, so that the slower absorption of the glucose, so that you will have the hypoglycemic condition. So this is the moisture for the calculation purpose. Protein is also another thing which will also influence the glycemic index, and the carbohydrate. Carbohydrate portion, basically carbohydrate, protein, fat. These are the other three components which also influence the glycemic index overall. So we have screened these also, because when we have added recipe we are adding external fat. That's why how the glycemic index influences also, which we have seen along with the alpha amylase activity. So the dietary fiber, this is a very rich source of these varieties, particularly this bitter gourd which we have screened, and we have seen that the dietary fiber, particularly the soluble fiber, which is more, and this is the major another active molecule which also the matters for the glycemic index. So if you look at, this is the essence, basically the bitter gourd as it is, what is available in the market is only 54, even you look at 49. Actually recent meta-analysis, particularly for the type 2 diabetes, if you want to manage your diabetes through the diet, it is an advice that you should have the glycemic index will be around 49 to 50, 52, something like that. If you maintain your diet like that, you can easily maintain, which is possible through manipulating with bitter gourd in the various recipes, so that you could bring down your glucose levels in the body. So these are for those 4 varieties which I am giving, and this is the essence what we have worked. And finally, eat more foods that grow on trees and plants, and choose carefully foods that is manufactured in plants. Thank you.
Maybe we can have just one question.
Then it is not answered, ripe or ripe draft.
I have told you, ripe means it is totally changed the color, like fruit, you know what you call.
Which one is beneficial?
Raw only. Ripen has higher glycemic index because more sugars are formed. Alpha amylase inhibition activity also good.
In terms of anti-diabetic compounds.
In terms of anti-diabetic compounds also, alpha amylase inhibition also goes down there. That's why the glycemic index is gone up.
So raw is better.
Raw is better. Actually, here the practice also, people they don't eat totally once it becomes color.
Say the ripe one contains.
Yeah, because of the color, some of the molecules, particularly when the chlorophyll is gone and these anthocyanins and other things will emerge. But what quality molecules also very important. Now we are looking at the combination permutations also, because now we are playing with almost now 32. My target is 100. So let us see for the absorption, particularly bioavailability, how it will go with this raw, which I am interested. Only for an academic exercise which we have done with the ripened one.
Is there any evidence to correlate that your glycemic index lowering potential is correlated with the terpenoid in the bitter gourd, or is there any correlation with what can be found in glycemic index measurement and the hypoglycemic effect of bitter gourd?
So talking
One
about
small question, how can glycemic index be determined either in vitro?
glycemic index, how we will do it in vitro.
How can that be found by the in vitro?
Yeah, it's now actually established methods now, our lab. FAO WHO protocol in vivo methods have been established. Now NIN has, now even probably you might be knowing that the machines are also out now. UK Food Science and Technology Group has developed now in the market now, in vitro you can do glycemic index. Otherwise in vivo is a cumbersome, you need 10 volunteers, they have to come 4 times to your lab to test the product. Otherwise for in vitro you could simulate the conditions in the test tube. Even how many times which we will masticate the sample also, which we are mimicking through the centrifugation and by shaking water box also. It's exactly how it mimics in the human intestine will be mimicked in the test tube, and we will add the enzymes and we will take out and we will do the dialysis also. So that how much glucose is crossed from in the dialysis tube, then we will measure the glucose also. So it's a correlated and well established methods actually.